Hello everyone and welcome back to this new session. So myself, Dr. Narkhade Gopal Vasudev, working as Assistant Professor in Department of Genetics and Plant Breeding, School of Agriculture, SR University, Warangal. So today's topic of this session is breeding for value addition in crop improvement. So what is meant by value addition means what? That is breeding for improvement of quality traits in crops. So first we will see what is meant by quality. The quality refers to the suitability or fitness of an economic plant product in relation to its intended end use. So quality refers to the suitability or fitness of an economic plant product. The concept of quality is context dependent and varies according to the specific requirement at different stages, including seed quality, crop growth performance, final crop product characteristics, post-harvest technology, consumer preferences, cooking quality, keeping quality, that is storage, and transportability. So this concept of quality is context dependent. It depends on the context. Thus, the quality is not a single attribute but a composite of multiple traits determined by the purpose for which the product is used. Right. So quality is defined as degree of excellence of the product for that specific use. So in agriculture and plant products, the quality is multidimensional concept that includes several attributes such as color, size, nutritional content, shelf life, suitability for processing. So with all these are considered as the quality characters, several attributes of quality. Thus, the quality can be understood as a fitness or suitability of an economic plant product in relation to its intended use. A quality trait refers to the specific characteristics. A quality trait refers to a specific characteristics that determines the quality of the produce. So each crop possesses a distinct set of quality traits which are specific to the species and its intended use. So these traits vary among species and are largely influenced by the plant part utilized as economic product. So for example, seed, fruit, leaf or tuber. So thus the quality traits are crop specific, use oriented and multidimensional, forming the basis for selection and improvement in plant breeding. Then now we will see the classification of the quality traits. So the quality traits in crops can be broadly classified based on their nature and genetic control. So first, based on nature of traits, like first in that nature of traits, first is morphological traits like physical attributes such as seed size, shape, color. Then organoleptic traits, that is sensory qualities like taste, aroma and texture. Next, nutritional traits, that is composition of nutrients, protein, vitamins and minerals. Next, biological traits, so factors like shelf life, pest and disease resistance and storability. In others, the most important, that is processing quality and industrial suitability. So these are two are the main major traits based on the nature of use. Then second, based on the genetic control, oligogenic traits, oligogenic inheritance. So it is controlled by few genes and the traits are qualitative in nature. Polygenic inheritance means the traits controlled by many genes and those traits are having quantitative in expression. Third, maternal inheritance. So governed by cytoplasmic genes inherited from female parent. Now one by one we will see. First, morphological traits. So morphological traits are quality attributes related to external appearance of the produce. So they are primarily concerned with size, shape and color. So which are the key determinants of visual quality? So common examples will include grain or fruit size, grain or fruit color. So shape and overall appearance. So these traits are easily observable and measurable, making them important for the rapid assessment. Morphological traits play a crucial role in consumer acceptance, as the visual appeal often influences the market preference and purchasing decision. So that's why the visual appeal is one of the most important aspect which will influence the market preference and purchasing decision. Second, that is organoleptic trait. So organoleptic traits are quality attributes related to palatability and sensory perception of that particular produce. So they include characteristics perceived through human senses such as taste, aroma, smell, juiciness, softness or texture. So these traits are easily detected through sensory evaluation and are critical in determining eating quality. So organoleptic traits play a major role in influencing consumer preference. These organoleptic traits play a major role in influencing the consumer preferences and acceptability, often deciding the market success of crop variety. Next, nutritional quality. So the nutritional quality refers to the value of produce in human and animal nutrition. It encompasses key components such as protein content and quality, oil content and fatty acid composition, then vitamins and mineral content, and last, presence or absence of anti-nutritional factors. So unlike morphological or organoleptic traits, nutritional quality is not easily pursued directly by the consumers or farmers and often requires laboratory analysis. However, it is of paramount importance as it directly influences the human health, animal productivity and overall nutritional security. So that's why these nutritional quality traits are having its own importance. So next, biological quality traits. So this biological quality traits refers to the characteristics that determines the actual nutritive usefulness of the produce. So what is the biological quality traits means? These refers to the characteristics that determines the actual nutritive usefulness of a produce, often assessed through feeding experiments on animals. So these traits helps the value of food for human consumption based on biological responses. So key examples include protein efficiency ratio, biological value and body weight gain. So such traits are not directly visible or easily perceived by consumers or farmers and generally requires experimental evaluation. Right? However, they are extremely important as they reflect the true utility and effectiveness of the products in supporting human and animal nutrition. So in case of other quality traits, these category include quality traits that do not fall under morphological, organoleptic and nutritional or biological groups, yet significantly influence the overall utility of the products. So this category includes quality traits that do not fall under morphological, organoleptic, nutritional or biological groups, yet significantly influence the overall utility of the products. So the important examples includes cooking quality. For example, rice grain elongation, texture after cooking. Then second, keeping quality, shelf life and storability of fruits and vegetables. Third, processing quality, suitability for milling, baking or industrial use. Then fourth one, fiber quality. So fiber strength and length in cotton. So many of these traits are of prime importance as they are directly determine the economic value, usability and market acceptability of the products. So now the next is we will see the overview of quality traits in some major crops. So quality traits of selected crops will in first wheat. So amber grain color with medium to bold size, hard vitreous texture and lustrous appearance for better market quality, high lysine content and good baking quality, essential for bread and biscuit making. Rice. Next crop is rice. So wide, fine, long, slender grains are preferred. Desirable taste and aroma, that is fragrant types. Low breakage during milling and high hulling recovery. So superior cooking quality, higher protein and lysine content. Then next crop is maize. Bold flint grains with attractive color, high lysine, oil and sugar content. Preferred seed color, that is yellow or white. Sorghum, bold grains with thin pericarp, white grains with attractive shapes and size, high protein and lysine content. Next, pearl millet. So bold, lustrous grains with pearly umber color, high iron content, that is nutritional significance. Then in barley, in malting barley, low protein content is desirable. High extract of soluble oligosaccharides after malting. Low protein reduces the beer husk, while higher soluble sugars will aid to fermentation. Now next crop, pulses. So attractive shape, size and color of the grains. High protein content, improved amino acid profile, that is methionine and tryptophan. Reduce flatulence factors, that is better digestibility. Then oilseeds, attractive seed size, shape and color, high oil content and quality. So low anti-nutritional factors, higher proportion of unsaturated fatty acids, that is better nutritional value. Then sugarcane, moderate hardness and long internodes, low, that is optimum fiber content for efficient milling, high sucrose content and desirable sucrose ratio, good juice quality, that is clear juice and with purity, no mixture. Then next crop, cotton. Some quality traits will include fiber length, fiber strength and fineness, maturity and uniformity and fiber color. Next crop, that is tobacco. So in tobacco, short thin leaves with less branch ends preferred for cigars. Thin leaves suitable for pipe smoking. Thick leaves preferred for cigarettes. Nicotine content high for bidi, hookah and chewing, low for cigarettes. High sugar content improves the quality. Next, jute, kenaf and sun hemp. So some quality traits will include fiber length, fiber strength and fineness, color and luster, and freedom from knots and specks. So in case of tomato, attractive shape, size and color of tubers, good taste and cooking quality, thin skin and good keeping quality, and starch content is high. Then in case of vegetables, high vitamin and mineral content, good taste, palatability, desirable keeping and cooking quality. Then forage crops, high nutritive value, greater palatability and free from toxic substances. Then next, medicinal plants, high content of active principle, that is bioactive compounds. Breeding for nutritional quality will focus on enhancing the nutritive value of crop produce for human and animal consumption. So the major objectives will include first high protein content and improved protein quality. So emphasis on essential amino acids, for example lysine and tryptophan. Second objective, high oil content and improved oil quality. So favorable fatty acid composition, more unsaturated fats. Third objective, increase vitamin content, enhancement of vitamins such as A, B complex and C. Then fourth one, reduction of toxic and anti-nutritional substances, minimizing the compounds harmful to human health. Then next topic, that is nutrition and nutrients. So nutrition is the scientific study of food in relation to health. Nutrients are the chemical components of food that provide nourishment and are essential for growth, maintenance and proper functioning of the body. The major nutrients include carbohydrates, fats, proteins, vitamins, minerals and water. So good nutrition refers to the intake of balanced diet that supplies all essential nutrients in adequate amount. Malnutrition results from deficiency, excess or imbalance of the nutrients in the diet. Therefore, all the nutrients must be consumed in appropriate quantities and proportions to maintain the optimal health. Protein content and quality. The proteins are the essential component of human diet, required for growth, repair and maintenance of the body tissues. The proteins are organic macromolecules composed of long chains of amino acids linked by peptide bonds formed between the carboxyl group of one amino acid and the amino group of another. The nutritional quality of proteins depends on their amino acid composition, particularly the presence and proportion of essential amino acids. A total 21 amino acids are important in human nutrition, which are classified into essential and non-essential amino acids. So now what are the essential amino acids? That cannot be synthesized by the body and must be obtained through diet. Those are called as essential amino acids. And what are non-essential means? That those non-essential amino acids can be synthesized within the body. Thus, both quantity, that is protein content, and quality, that is amino acid balance, are critical for determining the nutritional value of food proteins. Now we'll see essential amino acids. As I have told you that essential amino acids cannot be synthesized by human body and must be obtained through diet. So these essential amino acids include methionine, isoleucine, leucine, lysine, threonine, tryptophan, valine, phenylalanine, histidine and arginine. These are the essential amino acids. Then what about non-essential? So non-essential amino acids can be synthesized within the human body and therefore do not need to be supplied through diet. And these includes cysteine, proline, glycine, serine, alanine, aspartic acid, hydroxyproline, glutamic acid, norleucine and tyrosine. The quality of protein is primarily determined by its essential amino acid content and balance. So certain amino acids that are present in limited amount in a protein source are termed as limiting amino acids, as they restrict the utilization of other amino acids. So common limiting amino acids include lysine, methionine, tryptophan, threonine, isoleucine and valine. So these amino acids are called as limiting amino acids. So see, essential amino acid deficiency in some vegetarian foods like cereals, limiting amino acids are lysine, threonine and sometimes tryptophan. So in pulses, methionine and tryptophan. Nuts and oilseeds, lysine. These green leafy vegetables, leaves and grasses, methionine. So these are the limiting amino acids in some food. Then we will see what is meant by vitamins. So vitamins are the vital organic compounds required in very small quantities for proper growth, metabolism and overall health. They play essential roles in body growth, maintenance and reproduction. Classification of vitamins based on solubility. Vitamins are classified into different types. Then first type, fat soluble vitamins. So this fat soluble vitamins class will include vitamin A, vitamin D, vitamin E and K. So these vitamins stored in body tissues, mainly liver and fat. Then next, water soluble vitamins includes B complex and vitamin C, not stored extensively, require regular intake. Then synthesis and sources. So vitamin D and folic acid can be partially synthesized in the body. Most other vitamins must be obtained through diet. Vitamin D is associated with fat-rich foods and sunlight exposure. Other vitamins are abundant in fruits and green leafy vegetables. So nutritional quality of cereals and pulses. So cereals are the major sources of carbohydrates and forms the staple diet in many regions. They contribute approximately 70% of total caloric intake in the human diet. Right. Then cereals provide about 50% of the global protein requirement, whereas grain legumes, that is pulses, contribute to around 20% and remaining 30% is derived from animal sources. The average protein content in cereals range from 10 to 13%. However, cereal proteins are deficient in essential amino acids, particularly lysine, tryptophan, threonine. So these are the some mutants affecting the protein quality in cereals in different crops. So here I have collected like first crops, then different mutants, their origin with ideal feature and remarks. Like this I have given like some, like if we'll take example, maize opaque to spontaneous with high lysine and tryptophan, and automatically it will reduce the prolamine. So likewise I have given for barley, sorghum. So next we will see legume protein improvement. So legumes contain 18 to 28% of protein, which is nearly double that of cereals. In pulses, proteins are mainly of two types, albumins and globulins. The major storage protein in grain legumes is globulin, accounting about 80% of the total seed protein. So in chickpea, globulin occurs in three forms. First, alpha, beta and gamma. In faba bean and pea, globulins are of two types, that is legumin and vicillin. The legumin is less soluble in salt solution compared to vicillin functionally. Legumin does not coagulate at high temperature, that is approx 95 degree, whereas vicillin will coagulate, influencing the cooking and processing quality. Then limitations and improvement of legume proteins. So legume proteins are deficient in essential amino acids, particularly methionine and tryptophan. So legumes also contain several anti-nutritional and toxic compounds, including protease inhibitors, hemagglutinin, lathyrogens, glucosides, goitrogens, cyanogens, metal binding factors and antivitamin factors. So protein fractions in legume seeds like albumin, water soluble, amino acid balance is balanced, and remarks like minor fraction less than 20%. And globulin, salt soluble, deficient in sulfur containing amino acids and having major factor almost 80%. Then alpha globulin, deficient in sulfur containing amino acid, 80% of the total globulin, and beta and gamma globulins are the having minor fraction. Then in protein fractions in legume seeds. So first legumin, less salt soluble, non-coagulating, approximately amino acid is balanced and more predominant in faba bean. Then next vicillin, more salt soluble, coagulates at 95 degrees centigrade. It is deficient in sulfur containing amino acid and is dominantly found in French bean. Then genetics of nutritional traits. These nutritional quality traits in crops are governed by different types of genetic control, which determines their inheritance pattern and ease of improvement through breeding. So these traits may be governed by first oligogenic inheritance. This oligogenic inheritance means controlled by one or few major genes, exhibit qualitative variation, easier to manipulate through selection and breeding. Example, high lysine mutants like OPEC2 in maize. Then next, polygenic inheritance. So it is controlled by many genes, that is polygenes, show continuous variation, strongly influenced by environment. Example, protein content, oil content and vitamin levels. Next, maternal effect. So the traits influenced by cytoplasmic genes are maternal tissue. Expression depends on female parent. And important in traits related to seed composition and endosperm. So here I have collected the information like crop species, quality character and inheritance pattern. Like in sorghum, high lysine character is governed by single partial dominant gene. In barley, high lysine content governed by one major gene plus several minor genes. In oats, protein content is having complex inheritance. Low protein is dominant over high. In case of maize, sunflower, safflower, seed oil content having additive gene action. Sesame, seed oil content additive genes with partial dominance. That is, low oil content is dominant over high. Then in rapeseed, erucic and eicosanoic acid, two genes with multiple allele. Then same turnip rape, single gene with multiple allele. Safflower, fatty acid composition, three major alleles at one locus. Then in case of tomato, high beta carotene, two major genes plus modifiers. In carrot, carotenoid content having complex inheritance. Then next that we will see sources of quality traits in crop improvement. First, cultivated varieties. Existing varieties with desirable traits. Easily usable in breeding program. So second source, germplasm lines. Diverse genetic stocks conserved in gene banks. Provide novel variability. Third, spontaneous or induced mutants. Natural mutations are those created using mutagens. These are useful for traits like high lysine and dwarfness. Right. Next source, somaclonal variants. So this variation arising from tissue culture can generate unique quality traits. Next source, wild relatives. Source of rare and valuable genes. For example, resistance, nutritional traits may require pre-breeding due to linkage drag. Next, transgenes. Genes introduced through genetic engineering enable targeted improvement. That is, example, biofortification. Now, here see wild relatives as a source of quality traits. So, in case of cotton, for fiber strength, Gossypium thornberry and Gossypium armorianum. For fiber fineness, strength and maturity, Gossypium anomalum. For fiber strength and fineness, Gossypium raimondi and Gossypium tomentosum. So in case of tomato, fruit color, carotene content, Lycopersicum hirsutum. For high vitamin C content, Lycopersicum pimpinellifolium and Lycopersicum peruvianum. In case of pigeon pea, high protein content means Ataylosia species. In soybean, high protein content reduce lipoxygenase glycine soja. Oats, high protein, approximately 27%, Avena sterilis. So these are some wild relatives which can act as a source of quality traits in those respective crops. Then breeding approaches for the quality improvement. First, evaluation of germplasm. So screening diverse germplasm collections to identify the desirable quality traits. It provides base material for breeding. So next approach, mutagenesis. So induction of mutations using physical, that is gamma rays, or chemical mutagens. Useful for creating novel traits. Example, high lysine mutants. Third, hybridization, that is crossing between genetically diverse parents. It combines desirable traits into single genotype. Next, interspecific hybridization, crossing between different species, introduces the traits from wild relatives, that is disease resistant quality traits. Then next, somaclonal variation, variation generated through tissue culture, a source of new variability for quality traits. Next, genetic engineering, direct transfer of some specific genes. It enables precise improvement, for example, biofortification or protein enhancement. Then breeding methods for improvement, in that breeding methods for quality traits are similar to those used for yield or other traits. So commonly used breeding methods are backcross method, which is used to transfer some specific quality traits into elite varieties. The next, pedigree method, selection through generations based on ancestry. It is effective for traits with moderate heritability. Then third one, single seed descent method, rapid advancement of generations with minimal selection, useful for quantitative traits. Then recurrent selection, repeated selection and recombination. It improves the population mean for that particular trait. For example, protein content. Then next, progeny selection. Selection based on performance of offspring. It will enhance the accuracy. Then mutation breeding. Creation of new variability for quality traits by using some specific mutagens. So now, applications of breeding methods for quality improvement. Crop-wise, we will see first barley. Backcross breeding used to develop high lysine lines. Limitations, small seed size and low grain yield. Then sunflower. Oil content increased from 30 to approx 50%. Achieved through modified recurrent selection. Safflower. Oil content improved from 37% to 50% by reducing the hull content. Maize. Oil content increased from 4.7 to 17%. Protein increased from 10.9 to 23.5 after 70 cycles of recurrent selection. And limitation, significant reduction in the yield. So these are some applications of breeding methods for quality improvement. Now, the next aspect, seed oil analysis. So seed oil content is estimated using nuclear magnetic resonance NMR or either near infrared reflectance NIR analyzer. So what is the importance in breeding? It facilitates early generation selection for oil content. It saves the time by allowing simultaneous evaluation and planting. Widely used in oilseed crops like sunflower, groundnut and soybean. What are the key features? Non-destructive technique. Seeds remain viable and can be used for sowing after analysis. Second, it is suitable for segregating population. It enables the screening of individual plants. Then third, rapid and high throughput. So about 300 to 400 samples per day can be analyzed. It is simple and highly accurate. Next, protein analysis. So grain protein estimation is commonly done using protein analyzer. So what is the principle? This based on infrared reflectance (NIR) spectroscopy measures absorption of radiation related to protein content. Sample preparation: seeds are grounded into flour before analysis. It ensures uniform and accurate measurement. What are the key features? Rapid method. 200 to 300 samples per day can be analyzed. High throughput and efficient, suitable for breeding programs. Estimation of components, individual amino acid, calorimetric method, microbiological method. Anti-nutritional factors determined using chemical methods. Conventional versus modern methods. Jaldal method and for protein and Soxhlet for oil. High accurate but time consuming. Modern NIR based methods. Faster, non-destructive and suitable for large scale screening. What is the relevance to this? Breeding? Enable early generation selection for protein content. Supports screening of large population efficiently. Essential for quality improvement program in cereals and pulses. Then occurrence of toxic substances like present in grain legumes, oilseeds, vegetables, fruits and forage crops. Examples include protease inhibitors, cyanogenic glycosides, alkaloids, goitrogens, etc. Impact on health: cause adverse effects in humans and livestock. Toxic forests reduce the animal health, productivity and growth. Breeding for low toxic substances. In some grain legumes, oilseeds, vegetables, fruits and forage crops, toxic substances are found. So these toxic substances have adverse effect on human and animal health. Feeding of forage with toxic substances will adversely affect the health of animal. Therefore, it is essential to develop varieties of forage and food crops with low level of toxic substances so that it should not have adverse effect when consumed by animals. Considerable progress has been made in the development of varieties with low toxin contents in above crops. Breeding for reduction in toxic substances requires lot of chemical analysis. Hence, development of simple, cheap, rapid and reliable methods of chemical analysis is essential. What are the practical achievements? Varieties with improved nutritional quality have been developed in several food crops in many countries. In common bean, seed protein has been increased from 21.9 to 24.6 and in soybean 42.8 to 46.1%. In sunflower, seed oil content has been increased from 32% to almost 50% and in safflower 37 to 50%. In maize, seed oil content has been increased from 4.7 to 17% and protein content from 10.9 to 23.5%. In wheat, Atlas 66 is an important source of high protein which is being used in breeding programs for improvement of protein content. Varieties with improved quality released in some crop plants in India. Maize, high lysine content, Proteina, Shakti and Ratan. Sugarcane, high sucrose content, Co671, Co6806, Co7314, CO7704 and CO62174. Barley, malting quality, current 15, current 92 and current 280. Lathyrus, low neurotoxin, USA 24. Soybean, high protein and high oil content, that is Li. Protein 43 to 45 and oil content 23 to 25. Rapeseed, high oil content K88 48.8% oil. Limitations, most are polygenic traits. It is difficult to estimate and evaluate, hence are more finances required. Low heritability and affected by environment. There is a negative association of seed protein with grain yield in both cereals and pulses. In some cases, improvement in quality leads to reduction in grain size and yield. Sometimes the quality characters is found in wild relatives or species. The analysis requires close cooperation of biochemists, which sometimes becomes limiting factor in the progress. Biotechnological tools that have been used widely in all the crops. So this is about breeding for value addition, that is breeding for quality traits in crop improvement. So here we have seen what is meant by quality, what are the quality traits, some crop wise quality traits with some examples, breeding methods, oil estimation, protein estimation with limitations. Hope you will find this session important. Right. So till next session, it's thank you very much.